Having just gone through some of the history of ecosystem ecology and where some of our concepts that we currently use had originally come from, we can now begin to understand some of the controls over ecosystem processes. And at this point, it's really important to acknowledge one more source of our ideas. In the textbook, the authors say that ecosystem structure and functioning are governed by multiple independent control variables. This is the theoretical framework that they use in the book, is to understand how these control variables independently determine how ecosystems function and how they are structured. This idea comes from Hans Jenny. It's taken from soil science. Hans Jenny was a pedologist. He was a soil scientist. Born in 1899, he lived to 1992. He lived to be 92 years old. And in 1941, he published a very important book. It's probably one of the most important books that you would read as an ecosystem ecologist. So whether you go to the library, whether you go to Amazon to pick it up, it's really important at some point to at least take some time and sit down and read Hans Jenny's book, Factors of Soil Formation. Because what Hans Jenny had done is spend his entire career trying to understand soils. And the information, the knowledge that he developed about soils is important, but the theoretical framework that he used to understand the variation that he saw in soils all across the world became some of that conceptual framework, the infrastructure that we use in ecosystem ecology. When Hans Jenny traveled throughout the world, he saw soils that were really different. He could go to the temperate forests of eastern US and see soils that had large, dark horizons up top. And they looked down below slightly different, less organic matter, different colors. But then you go to another place, if you were to go to southeastern US, for example, and dig a deep pit, your soils would look really different. And what is it that was structuring the differences among these soils? His idea that he came up with was state factors. And he said that when you look at soils, they can really be, the variation that you see in soils throughout the world can really be attributed more or less to 5 different factors. And he had an equation in his book. It said S equals a function of CL, O, R, P, and T. Often we'll abbreviate that and just say clorped. But what that means is that soils, the structure, the function of soils, is really dependent on 5 different things, 5 different state factors. The first is climate, the climate at a site. The second are the organisms that you see there, the relief, the parent material, and how long that soil has been developing. And his conceptual framework was essentially to say that these 5 factors affect soils independent of anything else that's going on. And if we want to understand the variation that we see in soils, you have to understand what the climate of the site is, what organisms are there, both plants and animals, for example, the relief, where it is on the landscape, and what gave rise to that soil, what was its parent material, and lastly, how long it's been there. And just like Hans Jenny had said that state factors affected soils, we can take that same theoretical approach and say that ecosystems, how they're structured, how they function, is really a function of state factors. The same state factors that determine soils can be used to understand the variation that we see in ecosystem structure and function globally. Now, a key idea for state factors is that it's outside of the control of local organisms. Essentially, it comes down from above or below, and there's really not much an organism can do to affect the climate of the site, or the parent material, or the time. Now, that's a little bit of an abstraction. We already know that organisms can impact the climate of the site, but by and large, they're more or less independent. And once we know that, we can begin to understand how those individual factors affect ecosystem structure and function by trying to hold all other things equal. So for example, if we want to understand how time since disturbance or time since an ecosystem was first initiated impacts the structure and function of ecosystems, we would use a chronosequence. So everything else, all the other state factors are held constant, and we just try and find contrasts in ecosystems that really only differ in time. Alternatively, we can try and understand climate, and this would be a Climosequence. So if we want to understand how temperature impacts ecosystem structure and function, we would try and find a gradient where climate varied, but more or less we had the same parent material in similar organisms, for example. Lastly, we might use a toposequence. We might go from the top of the hill down to the bottom of the hill to understand relief, for example, and how position on a landscape impacts how that ecosystem functions. As an example where this was first applied, I had talked about Coles in Chicago, in the University of Chicago, and his work off the shores of Lake Michigan at the Indiana Dunes. And essentially what Coles had done to understand succession was to do a chronosequence. So I alluded to this before, but essentially what Coles had done is that if you start here, you have sand, and as you work your way back, you're getting older and older and older beaches. So Lake Michigan used to be over here, and this is an old beach, slightly younger beach, even younger. And this sequence right here is primarily a chronosequence. And vegetationally, you can see as you go from the lake, you go through grasslands, shrublands, and into forests, and this represents time also. And you can take a look at how the ecosystem developed if you assume that a long time ago, what's currently a forest used to be a shrubland, and before that it used to be a grassland. So if we want to understand how ecosystems develop, so to speak, on a 10,000 year scale or a 1,000 year scale, we can't wait that long, but we can go and take a look at chronosequences to begin to help us understand how ecosystems might develop. Now the state factor approach, we're going to work on that in a little bit and expand that and apply it a little bit more directly. But it's important to understand that there are some factors for which there are no natural independent gradients. We can find gradients in climate, we can find gradients in parent material, we can find gradients in how long ecosystems have been present on a specific site, how old the soils are. But there are some factors that we want to understand as ecosystem ecologists that there aren't natural independent gradients. And these are places where experimental manipulations are really important. Experimental manipulations are important for other things too, but here we really have no choice. So for example, if we want to understand the impacts of elevated CO2 on plant growth, there really isn't a natural gradient to use today that gives us a gradient in CO2 concentrations that might represent the CO2 concentrations that we expect in the year 2100. Or to go back to the Pleistocene, for example, when they were a lot lower than today. So for example, this is an experiment that elevates the CO2 concentrations of the plants inside of this ring. So this is the Biocon experiment. It's in Minnesota, and this is called a FACE experiment, free air carbon dioxide enrichment. And each one of these tubes, depending on the direction of the wind, releases pure CO2. It mixes and then elevates the CO2 concentration inside of that ring. The wind blows from this direction, CO2 gets released from over here, and it goes and elevates the CO2 concentration throughout this entire ring. So this is a ring that has 550, for example, parts per million CO2, which is what we expect to be the CO2 concentrations in the future, for which there are no natural gradients. So sometimes we have to use experimental manipulations to understand how factors independently affect ecosystem structure, ecosystem function. So with that, it's really important to understand that often in ecosystem ecology, we need to use gradients to understand how processes impact ecosystems. And for that, we use a state factor approach to understand how there are 5 of the main factors independently impact ecosystems. So gradients are a really important part of what we do. Other times, we really have to rely on experiments. There are certain situations where it's relatively difficult to find gradients that we need or clean gradients that only manipulate a single factor. And in these cases, we also use experiments to understand other things that affect ecosystems for which there aren't natural gradients. The key thing with state factors is the idea of independence, that what we see, variation that we see in ecosystems, is impacted by these state factors, and there's a degree of independence. There's no interactivity there. So, to sum up, state factors, they're really important. As you'll see in the next section, it sets the structure, the intellectual infrastructure for how we think about ecosystems and how we compare them.